Berkeley.
All right, hi, we're UC Berkeley. I'm Samantha. That's Jennifer, Caitlin, Brian, Will, and Dan. And that's also Matt, but he's sick today, so we're going to let him take a rest. And in the spirit of iGEM, where all these communities are coming together and sharing information, we thought that we could make bacteria share information too. So here's our project on addressable cell-to-cell communication in bacterial networks.
So our overall goal for the project is to create a cell network that can pass DNA messages to each other through a predetermined line of communication. So as you can see up here, the first cell will send a message to its target cell, and in turn, that cell will send a message to its target cell, and so on and so forth, creating a line of communication. Every cell is assigned a specific RNA address, which is the basis of discrimination between cells of different communication lines. To make these networks, we first have to pass the DNA from one cell to another, and we do this by using bacterial conjugation. And then through riboregulation, we will specifically choose which cell can read the messages. We have 4 specific goals. We want to construct high-performance riboregulators, harness bacterial conjugation, transmit a coded message, and finally, create a bacterial learning network.
So to illustrate how exactly a riboregulator works, we'll first look at the standard strand of DNA. Shown here is a strand with a promoter, a ribosome binding site, a coding region for red fluorescent protein, and a double terminator. RNA polymerase recognizes the promoter, and it reads through the strand of DNA and creates a complementing strand of RNA, which is then read through by a ribosome. However, if we replace the upstream region of the strand with a locked riboregulator, the ribosome binding site is sequestered and the ribosome has no access to the ribosome binding site, and translation cannot be initiated. If we introduce our key structure, it binds to a region near to the ribosome binding site, and the lock unlocks. Then the ribosome has full access to the ribosome binding site, and it can create all the proteins necessary, in this case, the red fluorescent protein.
Sorry. Okay, so we started this project with the best BioBrick riboregulator at the time, which showed a 3-fold gain from locked to unlocked in activity. And so we wanted to improve this as much as we can. And our strategy to do this was to make slight perturbations to our existing system, and then take from that the advantageous modifications to build an even better system. The first avenue we explored was that on the lock side. And what we tried to do is increase the strength of the ribosome binding site on the lock, and we found that both increased the background fluorescence and the activation fluorescence, which proved to have minimal overall gains in the system. Similarly, we experimented with decreasing the strength of the hairpin in the lock, which did have a little bit more advantageous effects because it tended to keep the background fluorescence the same, whereas it increased the activation fluorescence in the functionality of the riboregulator. However, these effects weren't nearly as effective as modifications to the key side of the problem. The first avenue we had to explore when modifying the keys was this idea that you needed an imperfect match from the addressing sequence on the key to the complementary sequence of the ribosome binding site on the lock. And so what we did is we made a key with a perfect duplex just to experiment with this. And we found that having this perfect duplex or having slight mismatches in the addressing sequence was neither detrimental nor advantageous to the overall activity of our system. And so we stuck with this idea and just kept the perfect duplex because this was an easier scheme to pursue. However, secondary structure seemed to be much more critical in the ways of improving our system, and so we found that increasing the secondary structure in and around the addressing sequence of the key tended to correlate positively with increased activity or activation of our keys in the locks. So the first key we made was this one right here, which was just the addressive sequence by itself and no secondary structure. And this was detrimental to the overall system because this was unable to activate the riboregulator and unlock the lock. Whereas these more complicated secondary structures, which include the addressive sequence in the anticodon loop of a SER2-derived key, for instance, or the loop of a very strong hairpin, seem to be the most activating of the keys that we made. So we stuck with this, and we wanted to optimize this architecture. And one of the ways we did this was to vary the spacing between this addressive sequence and the stems of all these hairpins. And we found that the shorter these loops were, the better these keys were at unlocking our locks. Furthermore, we experimented with modifying the 3' ends of our keys, and we found that if you tack on like a large protein encoding sequence to the end of our keys, it killed the effect of the activation potential of our keys. Whereas if we found consistent twofold improvements over all our keys with the explicit presence of a double terminator. And so we were able to take a system from a threefold improvement, or a threefold gain in activation, to over an 80-fold improvement in activation, which was a lot more suitable for our needs in conjugation.
Okay, now that we had a great set of riboregulator lock and key, we needed a way to transport them. So we decided to use bacterial conjugation, which there are naturally occurring plasmids in nature, the F plasmid and the RP4 plasmids, which have the ability to transfer their own DNA into a recipient cell. These F plasmids and RP4 plasmids are really large plasmids. They're about 60 to 100 kb, and they have a lot of genes that are necessary for conjugation. However, we decided to focus on one site, the origin of transfer, which is right here. And this is a site where rolling circle amplification takes place so that it can feed the plasmid over to the receiver cell. We decided to focus on this genetic element because if you knock it out, it no longer transfers over. And however, you can complement ORET onto another plasmid, and instead this plasmid will be the one to move over. How we decided to assay conjugation was to use the kanamycin marker on the F plasmid and R plasmid, and also place a ampicillin marker on the ORET complement plasmid. And then that way, when we mated them and plated them on tri-can plates and tri-am plates, we could see which plasmids had transferred over and which plasmids haven't. So here are our results from these experiments. As you can see, the wild-type F plasmid has a very high conjugation efficiency, so we get pretty much a lawn on that plate. However, once we knocked out ORET, no more colonies show up on the plates. However, once you complement it, this is a very high bar, and it's almost achieved its original efficiency. We found approximately the same results for the R system, where the wild-type R plasmid is very efficient. However, once ORET is knocked out, you see nothing on the plates, and once again, once it's complemented, the conjugation starts working again. Now that we have conjugation under control, we try to see if we could have a cell send a coded message to a recipient cell. So how we decided to do this is we attached a riboregulator key onto the ORET complementation plasmid that we mentioned before. Then we had it conjugate with a receiver cell that carried a plasmid holding a lock RFP gene with a CAM resistance. So then we played it on CAM and AMP to select for cells that had both this one and this plasmid. And then we tested it for RFP fluorescence to see whether or not message. From this, we saw that riboregulators don't affect conjugation efficiency. The key with the ORET transfers just as efficiently with the ORET by itself. However, we also saw that the key needed the ORET in order to move into the next plasmid. Next, we also saw that conjugation does not impede riboregulator function. As you can see, when conjugated, the RFP fluorescence is just as high as if you just take the lock and key and co-transform them into the same cell. So now that we have a way to send a coded message over to another cell, we've tried to think of more complex applications such as like computational aspects.
So the NAND gate is particularly well suited for enabling complex logical circuits because NAND gates are considered universal gates. That is, any logical computation can be formed from various configurations of NAND gates. A single NAND gate has the output shown in the truth table here. As you can see, the output of a NAND gate is high except in the presence of both input signals, in which case the output of a NAND gate goes low. Here is our design for a cellular NAND gate. The inputs to our output cell are a key and a locked lac repressor. The output cell has expression downstream of a lac promoter, such that when the lock and key are both present in the output cell, the expression is repressed, and thus the activity in the cell goes low. In any other scenario, the expression is constitutively on. As we design additional unique and independent locks and keys, we'll have additional NAND gates and potentially the ability to create more complex cellular networks.
So with the NAND gate, we have a universal computing platform. In theory, any computable function can be implemented by chaining together a number of NAND gates. Additionally, a bacterial culture has some other properties that are very interesting. We can get greater response to input, we can get positive and negative signals, and we can get a large number of signals occurring in parallel. These are the same properties that can be used to implement what's called an artificial neural network, which is another computational framework. We achieve greater response by altering the concentration of cells within our population. We tie the receipt of locks and keys to antibiotic markers, which allow us to enrich specific cell lines within the population. In this example here, cell 2 receives key 2, which unlocks an antibiotic marker, allowing us to enrich the concentration of cell 2 inside our population. Cell 3 does not receive a locked key pair. It does not unlock its antibiotic marker, and so we do not enrich the concentration of cell 3. Therefore, GFP is produced by cell 2, and the expression of GFP will be greater than the expression of RFP. By varying the number of key 2s that are in the population, we can then essentially change the weights of the connection between cell 1 and cell 2. This allows us to both get greater response to input signals and modify the weights of connections between signals. Using this, we can go on to build more complicated networks. For instance, we can use alternating layers of R and F plasmid types to build hierarchical networks where we can control the flow of information in a very fine-tuned manner. In this example here, we have what we're calling input signals that are tied to positive feedback mechanisms that propagate down the network. We then have what we're calling error signals, which are the negative feedback mechanisms that propagate up the network. This is what's known in computer science as the backpropagation neural network algorithm, and this is a method for implementing it in a bacterial culture. We'd like to thank the Arkin and Kiesling Labs for allowing us to work over the summer, along with Microsoft for providing funding. We'd also like to thank Lawrence Berkeley Laboratory and Jonathan Goller, Melissa Lee, and our wonderful postdocs, John Duber and Chris Anderson. And we'll now open up the floor to questions.
Very nice. Did you get any sense of the speed of signal propagation?
I'll take it. So okay, the speed of conjugation is on the order of roughly 10 minutes or so, so the speed of propagation through the network will be on the order of minutes.
Do you know how different two riboregulator sequences have to be to stop them from crosstalk?
Well, as I showed in the previous slides, like you don't need a perfect duplex between the key and the lock. So if you have one, like for instance, we had a key that was maybe 3 base pairs off from a perfect duplex. So the perfect duplex would work, and so would this. But as you increase the space that the key binds to the lock, you increase the combinatorial space at which you could vary these sequences. And so in theory, you could have you could come up with a lot of locks and keys that don't crosstalk because you could have drastic differences in the sequences. And so maybe like modifying 1 or 2 base pairs is going to allow crosstalk, but you won't need to get that finer resolution between these because you have so much space to deal with.
How big were the much space do you have?
You mean of the duplex? The ones that we were using were on the order of 30 base pairs. So
did you say that the NAND gate was made in... I wasn't sure. The NAND gate logic, was that implemented?
The NAND gate was proposed, and we've shown that we have all of the tools in place to build it, but it has not been built and shown to work.
How kind of digitally does it have to work for it to be useful? Does it have to be, can it kind of work and still be useful for this application?
Right, so the point of the NAND gate is actually just to prove that the network is Turing complete. It doesn't actually, the implementation of the neural network uses greater response, so there is no actual NAND gate being used in that. And so the implementation of the NAND gate just shows the theoretical completeness of the neural network system.
So what do you think would be more limiting for scaling this up? The number of pieces that are available now for building various NAND gates, or the number of plasmids that are available for sending separate signals?
Right, so it's almost certainly the number of plasmids, and there are some issues that we have to do with essentially using things like superinfection or the PR116 in order to control the plasmid copy number and make sure that we're using our plasmids and ORET sites efficiently.
How mutagenic is the conjugation process, and how likely is it that cheaters will just arise by eliminating the riboregulators?
Conjugation?
What was the question?
Cheaters and conjugation.
What was the question?
Mutate.
So to the extent that that conjugation is mutagenic, as you propagate through your network, the easiest way to cheat is to eliminate riboregulation during mutation and transfer.
So we found that actually knocking out genes and mutating it so that it doesn't conjugate is actually pretty difficult. Like, it took us quite a few tries with the Dotsenko-Wanner method to get rid of the ORET. So I find it difficult to imagine that it could just get rid of the riboregulator function so that it wouldn't, I don't think it would be really a problem.
that's kind of like the whole premise of having locked messages. Like, these things will, maybe they will conjugate, but if it's locked, it's not going to do anything. So you need specific conjugates to happen in order to get any activity out of the system.
Unless it mutates so that you no longer need the key.
I suppose if your locks fail, though, then everything falls down the chute. So.
Yeah.
But
they won't fail.
We don't know. Well, we have no idea.
Yeah, that's true.
Yeah. So far, we haven't come across a lock that has just spontaneously failed. So we think it's pretty reliable.
Who believes in evolution?
Do you have any idea, or you mentioned some real-world application of this traveler network of cells?
Right, so okay. I guess probably the easiest one is that you can then build sort of adaptive things so that you can have essentially hardware that is able to add additional nodes to its network as it grows. So you're not limited by what you start out with in the situation. So you could have a small sort of seed population, which if it's finding itself in a very computationally intensive environment can grow. Another really interesting thing would be actually tying it to sort of environmental stress mechanisms, tying it into one of the glucose pathways or something like that, so that you can then use actual, not just artificial inputs of locks and keys, but actually have it respond to other stimulus that's going on, other pathways that are going on in the cell, which would be somewhat interesting, giving it then sort of a network of bacteria the ability to compute things based on its environment.
There must be some parallels with natural ecosystems, maybe not the conjugation system itself, but the strengthening and breaking of the gradient response between cells by cell-cell communication.
Right, so this kind of parallelized distributed learning is thought to occur quite often in nature.
Keys and locks that don't have any crosstalk. So I was just wondering what your thoughts on that were.
So let me get this straight. You think that the complementary sequence has to match the RBS perfectly?
Well, not necessarily perfectly, but it has to match. I mean, there's a DNA star sequence can serve as an RBS, which means that there's probably some constraints on what sequence your lock can be, which thereby constrains your keys.
Okay, yeah, so the way we got around that idea is, so you have this complementary sequence that binds onto the RBS, but that doesn't limit how much sequence upstream of that you can have. And so you could increase this space and then make your keys bind more to the sequence upstream, which doesn't matter. And you're gonna get much more selective binding through that scheme, because that sequence doesn't matter and you're not altering the RBS at all. So, any others?
To actually make this functional, I'm not strong in biology, so but would you actually need a way to remove the key to return it to its locked state to make this a functional network?
Right, so we can send multiple keys into a cell, so we can also kill cells, which is the easiest way of turning them off, essentially. And so, no, quite honestly, I mean, essentially one of the best negative signals is just constitutive cell death, which can also be locked, and so we can just have a positive key that goes in and does something, then we can have another key that goes in and kills that cell. And that's really the easiest way to handle that problem.
Okay, well, thank you very much.
Control scheme.